So, dear students, concerned with this anatomy of flowering plant, initially we have come across definition of cell, tissue, followed by that I discussed classification of the plant tissues. Followed by that, previous class we have come across secondary growth in dicot stem. Increase in the diameter of the plant body, increase in the diameter of the plant body due to addition of secondary permanent tissues by the activity of vascular cambium and cork cambium in the stellar and extrastellar region is called as secondary growth. Increase in the diameter of the plant body due to addition of secondary permanent tissues by the activity of vascular cambium and cork cambium in the stellar and extrastellar region is called as secondary growth. Based on the definition I put forward, secondary growth will occur both inside the cell and outside the stellar region. When they will ask separate questions, explain secondary growth in stelae. When they will ask the question, explain the secondary growth in stelae or stele. We have to write first point, intrafascicular cambium. Second, interfascicular cambium, stellar cambium, formation of secondary xylem and secondary phloem, and lastly we have to explain annual rays. When they will ask the question, explain extrastellar secondary growth or explain secondary growth in cortex, you have to explain formation of cork cambium, second formation of cork and formation of secondary cortex. So lastly, we have come across bark. All the tissue which is present outside, all the tissue which is present outside the vascular cambium is called as bark. It includes secondary phloem, secondary cortex, cork cambium and cork. Followed by that you have come across function. Bark will protect the stem or bark will protect the plant from the airborne fungal spores. Followed by that, so today I will switch over to lenticels, and this question may appear for 1 or 2 marks. Accordingly, you have to adjust the matter. So now look over lenticel. Before switch over to this lenticels, please take care of the spelling. Spelling is Lenticel, not double L. Here, lenticels, Lenticel. Look over the first point. Lenticels are lens-shaped. Lenticels are lens-shaped raised spots. Lenticels are lens-shaped raised spots developed on the stem. Lenticels are lens-shaped raised spots developed on the stem, especially during secondary growth. Lenticels are lens-shaped raised spots developed on the stem, especially during secondary growth. The question arises how they are formed. Look over here. At certain regions, cork cambium, or you can call it as a phellogen, cut off closely arranged parenchymatous cells on the outer side instead of cork cells. These parenchymatous cells soon rupture the epidermis and forms lenticels. Look over here, first point. Lenticels are lens-shaped raised spots developed on the stem, especially during secondary growth. Now question arises how they are produced. At certain regions, the cork cambium or phellogen will cut off closely arranged parenchymatous cells on the outer side instead of cork cells or instead of phellum. and these parenchyma cells soon rupture at the epidermis region and forms lenticels. And look over and take care of this point. At certain regions, the cork cambium or phellogen cut off closely arranged parenchymatous cells. Closely arranged parenchymatous cells on the outer side instead of cork cells or instead of phellum, and these parenchymatous cells soon ruptures the epidermis and forms lenticels. I will repeat and take care because it is a little bit complicated. Okay, so first point, lenticels are lens-shaped raised spots developed on the stem, especially during secondary growth. How they are produced? At certain regions, the cork cambium will cut off. At certain regions, cork cambium or phellogen will cut off closely arranged parenchymatous cells on the outer side instead of cork or phellum. These parenchymatous cells soon ruptures the epidermis and forms lenticels. Okay, second point. They are produced during the formation of periderm. They are produced during the formation of periderm. In my previous class, I have come across this word periderm. Phellum, phellogen, phelloderm together constitute periderm. As already I covered, peri means surrounding, derma means layer. Phellum plus phellogen plus phelloderm together constitute periderm. So lenticels are produced during the formation of periderm. Lenticel is filled with loosely arranged lenticels are filled with loosely arranged thin-walled parenchyma known as complementary cells. So take care of this third point. Once in the NEET examination, they have asked this question: where do you find complementary cells? Look over here. Lenticels are filled with loosely arranged thin-walled parenchyma known as complementary cells. Lastly, function. Lenticels are also called as ventilatory structures. Lenticels are also called as ventilatory structure because it is meant for aeration, or it helps the exchange of gases between the environment and internal tissue. Lenticels are also called as ventilatory structures because they are specially meant for aeration, or it helps in the exchange of gases between the environment with that of the internal tissues. In addition to that, they are also involved in transpiration. Already I have come across this word transpiration in my previous class. Loss of water in the form of water vapor, through the aerial parts of the plant body is called as transpiration. Loss of water in the form of water vapor through the aerial parts of the plant body is called as transpiration. Okay, don't bother about this word because the detailed matter is there in the chapter transport in plants. Look over the second point. Lenticels are produced especially during the formation of periderm. Peri means surrounding, derma means layer. Phellum plus phellogen plus phelloderm together constitute periderm. Third point, lenticel is filled with loosely arranged thin-walled parenchyma known as complementary cells. Last character, or instead of that you convert it into a function. Lenticels are specially meant for the aeration, and it is also involved in transpiration. Please remember, for CET purpose, lenticels are also called as ventilatory structures because it helps in the exchange of gases between the environment with that of the internal tissues. So that ends lenticels. So dear students, now you have come across only one internal structure, that is internal structure of the dicot stem. Followed by that I covered secondary growth in a dicot stem. Look over the way of adjustment of the matter. First I covered internal structure of the dicot stem. Followed by that you have come across secondary growth in dicot stem. Followed by that, so today you are going to study internal structure of the monocot stem. Internal structure of the monocot stem. Followed by that, I will switch over to internal structure of the dicot root. Followed by that, you are going to study secondary growth in dicot root. Followed by that, waiting, three more internal structures are there. Okay. Now look over internal structure of the monocot stem. Example, any grass plants. Internal structure of monocot stem. Example, any grass plant. When you take the transverse section of a young monocot stem, it will reveals all the anatomical details from periphery towards the center. Actually, this is ground plan. Okay, this is ground plan of monocot stem, out of which they have expanded only 25% of the figure. Look over, this is an ground plan of monocot stem, out of which they have expanded only 25% of the figure. I will explain the matter from periphery towards the center, not from center to the periphery towards the center. So when you take the transverse section of a monocot stem, you will get three regions. Number one, epidermis. Second, hypodermis. And third is ground tissue. The question may arise for 5 marks. The way of arising the question, explain the internal structure of the monocot stem. At that time, figure will carry 2 to 2.5 marks. Explanation will carry 2.5 or sometimes 3 marks. Instead of that, they may arise, draw a neat label diagram of anatomy of or TS of monocot stem. Only figure will carry 5 marks. Look over the way of arising the question. When they will ask the question, explain the internal structure of monocot stem. Figure will carry 2 or 2.5 marks, 2.5 or 3 marks for the explanation. Instead of that, when they will ask the question, explain, you have to write the concerned matter. Same way, when they will ask, draw a neat label diagram of, TS of monocot stem, or to draw only figure, explanation is not required. So when you take the transverse section of a monocot stem, you will get three regions, namely epidermis, hypodermis and ground tissue. First look over epidermis. Epidermis is the outermost layer. It is made up of nearly rectangular shaped cells. Epidermis is the outermost layer. It is made up of nearly rectangular shaped cells. Cells are tightly arranged or compactly arranged without any intercellular space. Epidermis is the outermost layer. It is made of nearly rectangular shaped cells. You know, in mathematics, rectangular means it is a geometric figure with 4 sides. Rectangular means it is a geometric figure with 4 sides. Rectangular shaped cells. Cells are tightly arranged or compactly arranged without any intercellular space. Outer wall of the epidermis will show a thin waxy coat known as cuticle. Outer wall of the epidermis will show a thin waxy coat known as cuticle. Cuticle will check the rate of transpiration. It not only gives protection but also checks the rate of transpiration. You know already, loss of water in the form of water vapor through the aerial parts of the plant body is called as transpiration. Look over the figure carefully, you will get one more idea. Trichomes are absent or stem hairs are absent. Okay, so that ends epidermis. Epidermis is the outermost layer. It is made up of nearly rectangular shaped cells. Cells are tightly arranged or compactly arranged without any intercellular space. Outer wall of the epidermis will show a thin waxy coat known as cuticle. Cuticle will check the rate of transpiration. Just below the epidermis, hypodermis is present. Just below the epidermis, hypodermis is present. It is made up of sclerenchymatous cells. Just below the epidermis, hypodermis is present. It is made of sclerenchymatous cells. You know already, sclerenchyma is a dead tissue at maturity. So the main function of this hypodermis is to provide mechanical support. The name hypodermis is derived from Greek. Hypo means below, derma means layer. Just below the epidermis, hypodermis is present. It is made up of sclerenchymatous cells. It is known fact that sclerenchyma is a dead tissue. Main function of the hypodermis is to provide mechanical support. Next is ground tissue. All the tissue which is present within the hypodermis is called as ground tissue. All the tissue which is present within the hypodermis is called as ground tissue. Look over the linings. All the tissue which is present within the hypodermis is called as ground tissue. So ground tissue is composed of parenchyma cells. It includes parenchyma cells. Cells are loosely arranged with intercellular space. Okay. All the tissue which is present within the hypodermis is called as ground tissue. Ground tissue is composed of parenchymatous cells. Cells are loosely arranged with a lot of intercellular space. You know already, main function of the parenchyma is storage of food. Main function of the parenchyma is storage of food. Next matter is related to vascular bundles. You know already, the compact mass of xylem and phloem is known as vascular bundles. The compact mass of xylem and phloem is known as vascular bundles. Here, vascular bundles are conjoint. Here, the vascular bundles are conjoint and closed type. The compact mass of xylem and phloem is known as vascular bundles. In case of monocot stem, vascular bundles are conjoint and closed type. Conjoint means xylem and phloem will lie on the same radius. In conjoint, xylem and phloem will lie on the same radius. Closed means cambium is absent. When cambium is absent, there is no chance for the secondary growth. Previously, we have come across cambium will play a significant role in secondary growth. So, in case of monocot, secondary growth is absent. Vascular bundles are conjoint and closed type. Conjoint means xylem and phloem will lie on the same radius. Closed means cambium is absent. Okay, in addition to that, you have to take care of some points related to vascular bundles. In case of dicot stem, vascular bundles are arranged in the form of ring. In a dicot stem, vascular bundles are arranged in the form of ring. But here, vascular bundles are scattered all over the ground tissue. Vascular bundles are irregularly distributed all over the ground tissue. So this is called as atactostele. Atactostele or atactostele. Vascular bundles are scattered all over the ground tissue. This kind of arrangement is called as atactostele or atactostele. But generally, smaller vascular bundles are present towards the periphery. Smaller vascular bundles are present towards the periphery, and larger vascular bundles are present at the center. Look over vascular bundle once again. The compact mass of xylem and phloem is called as vascular bundles. In monocot stem, vascular bundles are conjoint and closed type. Conjoint means xylem and phloem will lie on the same radius. Closed means cambium is absent. But this matter you should not write when they will ask monocot stem internal structure. Only you have to mention. Explanation is not required. So vascular bundles are scattered all over the ground tissue. This kind of arrangement is called as atactostele or atactostele. Smaller vascular bundles are present towards the periphery. Smaller vascular bundles are present towards the periphery, and the larger vascular bundles are present at the center. Smaller vascular bundles are present at the periphery, and larger vascular bundles are present at the center. Vascular bundle is surrounded by a bundle sheath. Vascular bundle is surrounded by a bundle sheath, which is made up of sclerenchyma. Vascular bundle is surrounded by a bundle sheath, which is made up of sclerenchyma. So take care of all the matter related to vascular bundles, because it will play a significant role in the CT. Vascular bundle is surrounded by a bundle sheath, which is made up of sclerenchymatous cells. Inside this bundle sheath, phloem is present on the outer side and xylem is present on the inner side. Inside the bundle sheath, phloem is present on the outer side and xylem is present on the inner side. Xylem is composed of two metaxylem and few protoxylem. So in addition to that, you have to remember water containing cavity. Okay? It is composed of two metaxylem and few protoxylem. So just beneath the protoxylem or just below the protoxylem, you will get water containing cavity. It is produced due to disintegration of one or more protoxylem elements during the elongation of the stem. Look over here. Inside the bundle sheath, you will get phloem on the outer side and xylem on the inner side. Xylem is composed of two metaxylem and few protoxylem. Just below the protoxylem, you will get water containing cavity. It is produced due to disintegration of one or more protoxylem elements, during the elongation of the stem. Water containing cavity is also called as lysigenous cavity. Water containing cavity is produced due to disintegration of one or more protoxylem elements during the elongation of the stem. Okay, so that ends internal structure of the monocot stem taken at the internodal region. Remember, this is an internal structure taken at the internodal region. Back to morphology of flowering plant, where you have come across nodes and internode. Node is the solid region. From which the leaf arises. Node is the solid region from which the leaf arises. So the distance between two nodes is called as internode. This is an all explanation related to transverse section of the monocot stem taken at the internodal region. I hope that node and internodes are clear. Node is the solid region from which the leaf arises. The distance between two nodes is called as internode. So previously, when you go through old CET papers, they have arranged this question for CET purpose. Okay, you have to go through this matter for CET purpose, but according to new NCERT, this much explanation is necessary. Don't go for the extended explanation of the monocot stem. Right. Now look over one more internal structure. Internal structure of the dicot root. Followed by this internal structure of the dicot root, you are going to study secondary growth in dicot root. Followed by that monocot root. Then you are going to study dicot leaf and monocot leaf. Followed by that, I will discuss few differences. Okay, now take care of the internal structure of dicot root. Example, sunflower. I hope that you know sunflower is not a flower, it is an inflorescence. Sunflower is not a flower, it is an inflorescence. Please remember, sunflower is not a flower, it is an inflorescence. So when you take the transverse section of a dicot root, when you take the transverse section of a dicot root, you will get three regions. When you take the transverse section of a dicot root, you will get three regions, namely Epibleum, number 2, Cortex, number 3, stele. So when you take the transverse section of a monocot root, or it may be a dicot root, you will get three regions. When you take the transverse section of a root, it may be a dicot or it may be a monocot, you will get three regions, namely Epibleum, Cortex and stele. First look over Epibleum, then I will discuss Cortex. Epibleum is the outermost layer. It is made up of barrel-shaped cells. Cells are tightly arranged or compactly arranged without any intercellular space. Epibleum is the outermost layer. It is made up of barrel-shaped cells. Cells are tightly arranged or compactly arranged without any intercellular space. Cuticle and stomatas are absent. Please remember, cuticle and stomatas are absent. But some of the Epibleum cells are drawn into unicellular root hair. Some of the Epibleum cells are drawn into unicellular root hair. It absorbs water and mineral salts from the soil. Epibleum is the outermost layer. It is made up of barrel-shaped cells. Cells are tightly arranged or compactly arranged without any intercellular space. Some of the Epibleum cells are drawn into unicellular root hair. Some of the Epibleum cells are drawn into unicellular root hair. It absorbs water and mineral salts from the soil. Next region is cortex. The region present in between Epibleum and stele or stele is called as cortex. The region present in between Epibleum and stele or stele is called as cortex. Okay, so it is made up of parenchymatous cells. The region present in between Epibleum and stele is known as cortex. It is made up of parenchymatous cells. Cells are loosely arranged with intercellular space. Cells are loosely arranged with intercellular space. You know already, main function of the parenchyma is storage of food. Main function of the parenchyma is storage of food. But in addition to that, it helps in the radial conduction of water. This parenchyma cells helps in the radial conduction of water. It helps in the radial conduction of water. Okay. The region present in between Epibleum and stele is known as cortex. It is made up of parenchymatous cells. Cells are loosely arranged with intercellular space. It helps in the radial conduction of water. In addition to that, main function of the parenchyma is storage of food. Next is endodermis. Please remember, endodermis is not a separate constituent, but it is the innermost layer of cortex. Endo means inside, derma means layer. Innermost layer of the cortex is called as endodermis. Endodermis is the innermost region of cortex. It is also made up of barrel-shaped cells. Cells are tightly arranged or compactly arranged without any intercellular space. Look over here. Endodermis is the innermost layer of cortex, or it is the innermost region of the cortex. It is also made up of barrel-shaped cells. Cells are tightly arranged or compactly arranged without any intercellular space. Cells are compactly arranged without any intercellular space. Most of the endodermal cells will show Casparian thickenings. Most of the endodermal cells will show Casparian thickenings. Most of the endodermal cells will show Casparian thickening. That is nothing but deposition. That is nothing but deposition of suberin. Most of the endodermal cells will show Casparian thickening. That is nothing but deposition of suberin. You know already, cork or phellum, where I covered cells are compactly arranged. That story. In addition to that, it is having suberin, a water impermeable material. Water impermeable material. So take care of this line. Most of the endodermal cells will show Casparian thickening, but some of the cells which are present just opposite to the protogelum, some of the cells which are present just opposite to the protogelum do not show this Casparian thickening. Such cells are called as passage cells or transfusion cells. Look here. Most of the endodermal cells will show Casparian thickening. This layer is so named because it was first discovered by the scientist Caspar. So as a mark of honor, it is called as Casparian thickening. Most of the endodermal cells will show Casparian thickening. That is nothing but deposition of suberin, a water impermeable material. But some of the cells which are present just opposite to the protogelum, some of the cells which are present just opposite to the protogelum do not show this Casparian thickening. Such cells are called as passage cells. Such cells are called as passage cells, or you can call it as a transfusion cells. Now question arises, what is the function of this passage cells? It will enable the entry of water. It helps in the entry of water from cortex to the stellar region. It helps in the entry of water into the stele or stele. Okay, so that ends cortex. Next matter is related to pericycle, vascular bundles, conjunctive tissue and pith. So indirectly I put forward stele is composed of Next matter is steely or steel. It is composed of pericycle, vascular bundles, conjunctive tissue and pith. It is composed of pericycle, vascular bundles, conjunctive tissue and pith. First look over pericycle. It is the outermost layer of stele. Pericycle is the outermost region of the stele or stele. Pericycle is the outermost layer of the stele. It will perform a very important function. It helps in the formation of lateral roots and vascular cambium. Pericycle is the outermost layer of the stele. It will play a significant role in the development of lateral roots and vascular cambium during secondary growth. Lateral roots. Back to morphology. I hope that you have remembered this line. If the root develops from the radicle, then it is called as a taproot or primary root. If the root develops from the radicle, then it is called as a taproot or primary root. When the primary root produces branches, they are called as secondary roots. When secondary root further produces branches, they are called as tertiary roots. Secondary and tertiary together constitute lateral roots. If the root develop from the radicle, then it is called as a taproot or primary root. When the primary root further produces branches, they are called as secondary roots. When the secondary root further produces branches, they are called as tertiary roots. Secondary and tertiary together constitute lateral roots. Okay. So next matter is vascular bundles. So vascular bundles are radial. Vascular bundles are radial. Concerned with this figure, it is tetrarch. General root tetrarch to hexarch. 4 xylem patches are phloem patches. I will explain, don't worry. Vascular bundles are radial, tetrarch to hexarch, closed and exarch. Look over here, radial. In radial vascular bundles, xylem and phloem lie side by side. Xylem and phloem lie side by side or alternate with each other. Xylem, phloem, like this alternate with each other or they lie on different radii. Tetrarch to hexarch. Tetrarch means 4. Hexarch means 6. Okay. Here, concerned with this sunflower, it is tetrarch. 4 xylem patches, 4 phloem patches are present. Tetrarch to hexarch, general matter, but concerned with the sunflower, it is tetrarch. 4 xylem patches and 4 phloem patches are present. Closed means in between xylem and phloem, cambium is absent. Exarch. When protoxylem is towards the periphery, and metaxylem is towards the center, then this condition is called as exarch. Dear students, please remember this line. We should not explain all the matter in detail. Okay, only got to mention vascular bundles are radial, tetrarch, closed, exarch. Explanation is not required in anatomy. Next is conjunctive tissue. The parenchymatous tissue that lies in between xylem and phloem is called as conjunctive tissue. The parenchymatous tissue that lies in between xylem and phloem is called as conjunctive tissue. Last matter is pith. Pith is the center region of the stele. Pith is the center region of the stele. It is made up of parenchymatous cells. But please remember, in case of dicot root, pith is narrow, not broad. It is narrow. The parenchymatous tissue that lies in between xylem and phloem is called as conjunctive tissue. Pith is the center region of the stele. Pith is the center region of the stele or stele. It is composed of parenchyma cells. But pith is narrow in case of dicot root. But it is broad in case of monocot root. Okay.